So my talk at this meeting is twofold. So I'm going to have a portion of that which is discussing the technology involved around transcriptomics, which is looking at RNA expression profiles. And we want to use that looking at our matched tumor samples with the normal. So knowing that the normal samples across all of us as human beings, there's a great amount of variation. So we want to make sure that every patient has, in fact, a true normal comparator, which isn't based on a random group in a population, but it's based on an actual normal of the same cell type that you have your cancer arising in. In this case, it's lung cancer. So that's the first portion of that, is discussing the background on it. And then the second portion is presenting data from the phase 1. So there's an initial set of patients that we've had on there that we've had enough time to track them in terms of follow-up clinically. And now we're just going to see if we can make sense of some of the data that we've generated on those to match that up with what we've seen clinically. Background on the SPRING trial. So this is a triple combination trial that is using an immune checkpoint inhibitor and followed up with a CDK4/6 inhibitors. And what we're doing is we had essentially doing a phase 1, which is a toxicity and dosing, and we're looking at that triple combination across a series of patients that have been enrolled in this trial. And we're looking, primary endpoint is to see if there's toxicity or adverse events, and the secondary endpoint is actually looking at response rates. So we collect tissue from, like I said, mentioned before, is the tumor and the normal. And what I'm able to do then is utilize that information. We run them as one single batch at the end, retrospectively go back and see if we are able to predict the response rates that we would have from each of these individual patients. So far, so really literally produced the data a couple of days before, so I'm still putting a talk together, the exact finishes up. So what we are finding is that patients that didn't respond, makes a lot of sense, especially on the immune side of it. We're seeing that those markers weren't expressed. So we didn't go in with a priori knowledge, knowing that this is the particular patient that should or shouldn't respond, since the goal of this phase 1 study is to find toxicity. So I think what we did is kind of backed up what we've shown before in one of our earlier publications on the SIMS algorithm, that we definitely want to do a more prospective job of predicting patients and which combination therapies they would have. But we still see that some of these patients responded, and there's definitely rationale that we see behind it of the particular markers that these genes, that the markers of gene expression are showing that the patients that did respond would have been predicted to have that based on not necessarily the gene signatures, but the overexpression of these particular markers for these drugs. And patients that didn't respond, actually did not see a statistically significant expression difference. So the patients are actually enrolled. So the trial is set up that they do not have particular alterations, these particular fusions, RET, ALK, and EGFR fusions or mutations. And beyond that, these are metastatic lung cancer patients. And this is whether or not they do or don't fit into other certain criteria is based on judgment that gets together, outside of, with the WIN consortium. The executive panel decides all of that, and luckily those are things that the medical doctors get to do, and I just get to take the samples when they're sent to me. So far we've had 15 patients, but on the valuable side for me has only been 13 patients, and we'll be presenting data on 9 of those 13. So what's next is to, what we really want to do, instead of looking at sort of the single gene expression for these particular genes, we know that we, not just looking at this one particular triple combination, we know that we're looking at, multiple different combination therapies. We do believe in doing triple combinations to shut down multiple drivers of tumor progression all at one time, as opposed to doing kind of the traditional model would be, do one drug until it fails, and you kind of keep going in a stepwise fashion in series as opposed to in parallel. So we have 6 different triple combinations that we have identified that we are interested in doing, and what we'd like to do is really use the SIMS algorithm and utilize really the full expression profile instead of individual genes, and make sure that we can actually use that algorithm to correctly predict which one of the triple combinations we want to use in the future.